Okay, welcome everyone, and welcome everyone on Zoom as well. It's my pleasure to welcome you all to the thesis defense of Prashant Kalvapalle. He is a PhD student in the Systems, Synthetic, and Physical Biology program, and he has co-advised by Joff Silberg and myself. So before I hand it over to Prashant, I'm going to give just a brief introduction, not too brief introduction of Prashant and some of his work, but also what it's been like to work with him throughout his PhD. And this is on behalf of Joff and myself. So let me see how I advance these slides. There we go. So I just first want to say that Prashant is the first student that Joff and I have had the pleasure to co-advise together. And that's, for Prashant, that's meant he's had to deal with 2 different advisors, attend 2 different lab meetings, and really work at the intersection of environmental engineering and science and synthetic biology. And this is what I think of as kind of an emerging field, and Prashant has emerged as a leader in this space. And this is just a word cloud where I took his thesis document and giving you a preview of some of the things you're going to hear about today. But to point to some of his progress and success, Prashant has published numerous papers in his time at Rice. And I'll highlight 2 specific ones that are really focused on environmental synthetic biology, where Prashant has made a critical contribution. He'll talk more about these in his presentation today. But what does it mean to be an environmental synthetic biologist? It means using and developing tools from synthetic biology and applying them to study environmental phenomena and processes and shed new light on some of these really challenging problems that we think about in environmental engineering, from horizontal gene transfer to microbial communication signals in representative environmental systems. So you'll hear a lot more about that, but I can't introduce Prashant without also talking about just the massive impact that he has had in my lab, working on many, many different projects. So I'm highlighting here a lot of the other publications that Prashant is co-authored on, and specifically focus on wastewater monitoring. Without Prashant being involved in these projects, our massive wastewater monitoring system would not have been a success. And he's really kind of lent his expertise on many projects that everyone's worked on in the lab, and he's always willing to help out and help people figure out their issues in the lab. And so if I take kind of all the manuscripts that he's actually co-authored on and put a word cloud around that, you'll see he's an expert in much, much more than just environmental synthetic biology. Tons of expertise in wastewater, assay design, and much, much more. And I think if you were to talk to his lab mates, the way they would describe Prashant, in addition to being extremely knowledgeable and an amazing scientist and researcher, is as an extremely patient mentor and teacher. He is always willing to lend his time and help people figure out how to analyze their data, how to plot it, how to communicate it more effectively, how to design a new assay. If I look back at some of the Slack messages that Prashant has shared in our group, a lot of it is trying to share some of the resources that he's found that have helped him, and also develop new resources that other people in the lab can learn from. I love pointing to this. This is our Stouder Group shared resources file. And if you look at the creator of literally every single item, it is Prashant. From Zotero databases to YouTube videos on how to order probes from different vendors. He literally walks you through how to click through and orders these things, because everybody comes to him over and over and over. He realized it'd probably be better just pointing to the videos first. And these videos, on YouTube, you could look at them too if it'd be helpful for you, are on everything from things like primer design. And this one's been viewed 2,000 times. So clearly it's a resource, not just within our lab, but for other people, to really benign things like how to refill a pipette tip rack efficiently. Also been viewed almost 2,000 times. Anyways, these videos are truly a lasting resource and really just show how generous Prashant has been with his time in training others and helping other people learn new things. Another thing I'd like to mention is that Prashant has really been a champion for sustainability. He created our Sustainable Lab Slack channel. He's constantly reminding, not in a dogmatic way at all, but people to think about how they dispose of things, trying to think of new systems to make recycling and disposal of waste more easier and more sustainable. And so I love looking back at some of these messages she sent, where he's pointing to the boxes that we're throwing away in the wrong bins, and just reminding us in a nice way that we should be more conscious of how we dispose of things. After all, he is an environmental, synthetic biologist. And then finally, I think, I've already said this, the number one word I would use to describe Prashant is generous. He's generous with his knowledge, he's generous with his time, and that's really made him just such a joy to work with. So with that, I will hand it over to you, Prashant, and excited to hear.
Okay, thank you all for coming to my thesis defense. And a lot of people on Zoom. I'm really glad. I just sent the link to as many people as possible. So good, thanks for the good showing. I'll try to go a little basic on the introduction and I'll pick up speed in the middle. But if anyone has any questions either on Zoom or in person, please hold it till the end. And then we can take everyone's questions in the end. And then I won't keep you for too long. There's a lot of good snacks waiting for you outside as well. So my project that I'm going to talk to you about, as Lauren mentioned, with her great introduction, this is only a subset of work that I've been a part of. But this work is, I'm trying to focus your attention on the use of synthetic biology to modify DNA and RNA within bacteria, so as a way of recording information. And we do this not in ordinary lab bacteria, but microbes that I get from wastewater. Those are a little smelly and also harder to work with. So just a motivation, why would we want to sense the environment that microbes live in? And as you all may be aware of, microbes live in all sorts of environments. Within the human body is something that a lot of people are interested in, and a lot of popular knowledge is also there about. But soil has a lot of bacteria, and also wastewater is another environment where bacteria, their sole purpose in life is to eat away all the nutrients so that we can dump the dilute water into the river. So let's focus on soil for now. There's a lot of inputs that go into soil. If you're growing plants, you're a farmer, then you would deliberately add things to the soil that you think are helping the plants. But there's a lot of microbes within the soil. They're also seeing those nutrients. They're able to eat them and transform them into other things. By doing these tiny microbes have very outsized impacts. So they have very large ecosystem level effects, such as they influence how well your crops produce. So they influence agricultural productivity, but also by producing other gases, they impact climate change very heavily. And if you think about climate change can also impact the bacteria, which in turn amplify the effect of climate change even more. So that's all the more reason to study microbes within these environments, and also to use microbes to study what things here or on the left. So that is my goal. One of the examples of how big microbes can impact is if you add too much of nitrogen fertilizer, whatever your plants cannot use will be used by the microbes, and the microbes will convert it into nitrous oxide, also laughing gas, but it has 300 times the global warming potential of CO2. So if you're a farmer, you want to use my biosensor to check exactly how much nitrogen you're adding into the soil. So one thing we could do is either grab samples from the soil or wastewater, bring it into the lab and analyze it, but that's too boring. What we want to do is to use the microbes themselves as a way of sensing the environment, because naturally the microbes sense these things and they convert them into other things. So what we do is let the microbe give out another kind of signal that we can easily see and measure in the lab. So easier things to understand are visual outputs. So if a microbe glows whenever it sees, let's say, the fertilizer on the left, it's easy to figure out how much fertilizer there is by the intensity of the glow that you can see. But there's also other things that microbes can be made to produce, which are a lot more complex. But for example, if microbes can make a gas that is very rare in the environment, if you just put an instrument above the microbe, you can capture it coming out without digging in and disturbing the soil and the microbe itself. So the advantage of using a microbe as a sensor is that it can detect concentrations that are bioavailable. What that means is that there could be a lot of there could be a lot of the input variables in the environment, But if they're tightly bound to soil particles, then they can't be accessed by the microbes. Or if there's very little moisture, then things can't move around as easily in the soil. So that's the reason why if you use a microbe to sense what a real microbe would have been sensing, that's called bioavailable concentration. And also you get spatial information. There's not just one microbe, but there's many microbes distributed across space. So if you can read out all of them as a spatial scan, you can see that which subset location of your sample has more or less of these input variables. And also, like I mentioned before, if you dig the soil and take sample out, you're disturbing it. So if you use a microbe and you let the microbe do its own thing inside, then it's a non-destructive way of monitoring. So this is not the sensor that I work with, because there are a few caveats with this kind of sensing. So one of the things is, if you're using a visual method, which requires a microscope kind of a thing to look at your sample, you can't imagine putting a microscope into soil, because soil is a very opaque matrix. So light can't really pass through it as easily. So the microscope won't be able to see anything. The other part is, even if you are able to see the surface of the soil, if that's what you're interested in, you would require to have your visual device like the microscope constantly monitoring this sample, especially if you're interested in signals that are very transient. So let's say there's a rainfall event, happens for 30 minutes, and then you're interested in how much of the rain reaches the microbe and how much moisture they see, for example. So if you don't know when it's going to rain, you have to train your microscope, constantly monitor the sample over many days. So that is the kind of scenario we're interested in, monitoring things that happen very rarely over a very long time scale. So for this, rather than a microbe sensing things and producing immediate outputs, what we want to do is for the microbe to sense things and remember it. So we are calling this as memory. So these microbes that I engineer are especially relevant for long-term monitoring. So for example, if a microbe sees a particular rainfall event and then it records that it has seen a rainfall, we don't need to disturb the bacteria right then. We can wait for a week or two, and then we can see in the last 2 weeks, has a rainfall occurred or how much rainfall has occurred. So this is especially important for long duration monitoring. And also it can be used in other environments that I haven't mentioned here. For example, the human gut, which is like a set of tubes. Once bacteria goes in, you don't know what's happening to it until it comes out. It's similar to a network of pipes, especially if you don't want to break a pipe and take the microbe from inside to know what it's doing. You wait for it to come all the way in the outside, and then you can look at its memory that it has recorded while it was inaccessible. Also, I'm using DNA and RNA to record the memory. So the advantage is nucleic acids, which can be amplified very easily. And you can also do this from both cells that are alive and dead. And a tiny amount of cells is good enough to amplify as much as you want to get a good detection. So I'm dividing my talk into two sections. One of them is where I use the DNA as the medium to store the memory. And the second one is using RNA. So the DNA is appealing for long duration memory applications. So we all know that when a bacteria divides into two and then its daughters divide further, they faithfully replicate their DNA. So if you make any modification that tags onto their DNA, as long as it doesn't affect the organism in any way, they will just carry the DNA information that we are adding in faithfully. And the advantage with RNA memory is that it is distributed, it can be distributed within microbial communities because of this unique RNA sequence called 16S ribosomal RNA. It is there in all the organisms, but it has slight variations. So there's the loop region that you see here changes color based on which organism it is in. So because of this property, we can identify, we can put a memory onto these sequences, and we can distribute the memory, different memories within different organisms within the community. So this is appealing for monitoring microbial interactions. And I'll go a little bit more over what exact kind of interactions we are talking about. So let's start with the DNA memory for now. So we are interested in long duration environmental biosensing, and by recording whatever it's being sensed into the DNA. So the environment that we are interested in is not soil, it is actually wastewater. So what wastewater does is there's a high density of microbes that live in wastewater, and they're trying to eat away all the nutrients so that we can dump the leftover water into the river. So when there's a high density of microbes living anywhere, if you put in any materials into that, microbes tend to go and stick to those materials. So for example, if you had a filtering device in here and you want to filter the water for some reason, then your microbes will sit on the membrane and they'll completely clog it. So this is called membrane fouling. And this is very undesirable if you're operating a treatment plant. So every less than a week or so, you need to clear out the membranes or replace them very frequently. And it adds a huge cost to the treatment system. But some of the desirable properties could be, if you identify a subset of organisms within wastewater that are very important, let's say for transforming nitrogen away from nitrous oxide, but into nitrogen gas, which is a lot more harmless. So you could immobilize those specific microbes in either a plastic carrier membrane, or you could make them bind tightly to each other. So they do these things naturally. So our goal is to understand what they're doing and then try to nudge them, the desired organisms, to do the desirable behaviors. So the way that microbes do this behavior is when they are low in density, they try to talk to each other by secreting a certain compound called quorum sensing molecules. So this process is called quorum sensing, and the one example molecule is the one shown in the right. It's called acyl homoserine lactone. So I abbreviate it as AHL. It has 12 carbons. It's a huge tail, so C12AHL. So what they do is they all each try to produce, imagine, one molecule of this, and when they are huge in number, the density of the organism correlates with how much molecule they see. So at some point they turn on a switch-like response, and when that happens, they all together produce extra set of molecules that allow them to stick to each other and also stick to surfaces. So we understand how this process works within a pure microbe in a pure lab sample. But in a mixed microbiome like the wastewater, where there's many different kinds of organisms, we really don't know how this process works and which subset of organisms it works well in. So what is lacking here? The challenge is that we need to understand this quorum sensing in a wastewater community, and we need to understand it enough to be able to control it. So that's a long goal. So my project is where I deploy the memory-based biosensor within wastewater to detect this molecule that we're talking about. So let's say we can detect this molecule, how would we record its presence into the DNA? So that is the part I'm talking about here. So we are inspired from this natural system where when viruses naturally infect bacteria, sometimes they're lazy, they don't want to kill the bacteria right away. So what they do is they make an enzyme called recombinase, which cuts their own DNA and the bacteria's DNA as well. And so there are two different circles. When you cut them, they open up and they join together. So then you form this lysogenic life stage of the virus, where the virus is basically immortalized itself in the bacteria. So when the bacteria divides, the virus passively divides with it. It doesn't need to do anything. So that's the kind of goal we are going for, where we want to make a memory that's passively carried by the bacteria. So how do we exploit this? What we do is, instead of on two different molecules, we bring these two sites that the recombinase recognizes onto the same linear molecule of DNA. And when this cutting action happens, you can imagine there are 4 different ends that are produced. And depending on which end combines with which end, so if the leftmost end combines with the light red, you make a flipping event where the segment in the middle flips around. But if it combines with the rightmost segment, it forms a deletion event where the end segments come together, the part in the middle is kicked out or deleted. So we utilize both of these effects. Here I'm going to show a cartoon of how the flipping happens. So this is how we would make the flipping occur, or we would make the memory in the DNA. But how exactly does the sensing happen? I'll give you a very brief idea of how sensing works by looking at how gene expression works in bacteria. So this is called the central dogma of molecular biology. So there's DNA, and it has a gene that you're trying to make. So this gene will code for this orange enzyme. And there's regulatory regions, that is the promoter and another segment that I'll talk about in a bit. So when this molecule comes, the analyte that we're trying to sense, it binds to a transcription factor and activates transcription of this gene. So then the DNA becomes an RNA. And then the RNA has this ribosome binding site, which is carried over from the DNA. And then by recruiting other enzymes and other proteins, it forms the protein that finally carries the action in the cell. So what we do here is we are linking the sensing of this molecule to the production of this protein. recombinase that then makes the memory. So when we bring this all together, this is how it looks like. So this is the bacteria that's carrying our memory device in it. So there's the sensor module, which is triggered by the analyte. And then there's the reporter module, which is once the recombinase comes out, it binds to these two regions and then it flips around the segment in the middle. So I'm calling these two states off and on states. I designate them by different colors. So when the off changes to an on state, the information is stored in the DNA. But for us to be able to read this, there are two different methods that we can use. So how we store, so once the DNA is recording this information, there could be two methods. One is if you put a protein in the middle that flips around, you can make it turn on only in one of those two states. So you can use the fluorescence of the bacteria to figure out when the memory has been made. And so this is good because it's a clean transition from the previous sensors that I talked about that people are well versed with, and it's easy for them to use. But there's also a second generation readout, which is to use particular primer pairs here. And so the primer pairs are designed in such a way that only in the on state they amplify, but in the off state, they are not in an orientation in which they can amplify DNA. And for those of you who are not aware of what primers are, this is the same thing that people use to detect COVID. Like Lauren mentioned it in a lot of COVID monitoring work. So this is where the expertise came from. So whichever DNA or RNA you design these primers to target, that's the thing that they will specifically amplify, and you can detect it very easily. So the advantages with fluorescence is that we use a method called flow cytometry, and it can measure each cell at a time. So if you have a population of 1 million, 10 million cells, you get one value for each cell in the population. But with qPCR, we can get a quick, easy measurement, but you'll just get one measurement for the whole population. So there are pros and cons, but I like qPCR, so I'll recommend people go for that. So how exactly does the memory work? So before memory, this is how the signal would look like. Whenever the AHL molecule comes in, the sensor produces the fluorescence, and when the AHL molecule goes away, so the molecule is only seen in this dark gray band. And when the molecule is removed from the system, then the microbes forget that they have ever seen it. So there is no memory here. It's a real-time sensor. But if you incorporate the integrase, like I mentioned, so the integrase really flips the segment that makes the fluorescence, and then the fluorescence is actually permanent. So these bacteria at 48 hours, they actually remember what they've seen many hours before. So this is what I'm calling as the memory behavior. So within a single cell, memory is a binary system. So there's only an off state or an on state. But within a whole population, the way it works is if you put a tiny amount of things of the analyte, then a small fraction of population flips. And the more and more you increase this, the more of the fraction flips. So the real data is actually amazingly very similar to the theoretical cartoon I made. So the more and more of this analyte that I'm exposing the cells to, the more they flip. And the way I'm measuring this element on the y-axis is, this is the fraction of plasmid that is in the on state. So for that, I do a qPCR of these sequences that only target when they are in the on state. And there are two other primer pairs here. There's another primer pair that will target both the DNA that's in the off and on state. So this will give me a total count of DNA. This will give me the on, and by dividing one by the other, we get the fraction of plasmid that's in the on state. So one thing that's curious is at the highest concentrations that we expose them to, there's a slight dip in signal. That's because if you let them make too much of any particular enzyme that they don't need it for survival, they tend to slow down in growth. So this is called the burden of growth. So now what we want to do is bring everything together. We have a sensor that works in bacteria and the memory sensor. So we want to test it over long durations. So this is particularly the thing that people have never tested these sensors for. So for doing this, I have an assay that I've developed where this is very simple, easy to do. You just put this DNA sensor device into the bacteria, grow them up, and then keep diluting them with fresh food media, and then keep growing them every day. So that way you get as many generations of division as possible. And they are only exposed to the analyte for the first day, and then I let them grow for 8 more days after that to see how long the memory lasts for. So that is the experiment that I'm doing. And now you'll see the real data of how it actually works. So these two populations are the population on the bottom. The open circles are the ones that have never seen the molecule, and the closed circles are the ones that have been exposed to the molecule. And the element on the y-axis here is slightly different from previous graph. So here I measured the fluorescence using a single cell method. So when I do flow cytometry, I get a whole distribution of the population. And if I set a gate of where the negative population ends, everything above that gate, I'm considering it as the on state because it has a high fluorescence, and everything below it is the off state. So here again, I divide the fraction of the on by the total population to get the fraction that's shown in the y-axis here. So you can see that when they have not been exposed, they do have a tiny slight increase, which is unintended. So there's a leak from off to the on state. And when they have been exposed, as expected, they do turn on. So there's a high signal on day 0. But then as you proceed over time, it slowly goes back down to 0. So this device is undergoing some kind of a memory loss. So we are trying to understand why this happens. So both these effects, the off leaking and the on going away. So the leak is because even when there's no analyte that we are exposing them to, there's a baseline expression of this recombinase enzyme that makes the memory. So that's what turns a few cells into the on state. So in the other scenario, when we deliberately expose them to the analyte, they turn to the on state, but then they don't like being in that on state, especially because of the burden of making the fluorescence. What they do is the cells that are making fluorescence grow slightly slower than the cells that don't make anything. So because of that, when they grow over many generations, the tiny difference in cell in the growth rate affects them a lot. So they get outcompeted by the off-state cells. So they would either come back to the initial state in which their memory is completely lost, or they would go into a third state where there are like many more random mutations that occur in the DNA that we put in. And then these cells are not useful for anything. They can't sense the next pulse of the molecule they see anyway. So if you can do something to avoid these two problems, we can improve the memories sensor. So for tackling the first problem, what we do is we reduce the strength of the ribosome binding site, which reduces the amount of protein that gets made. And by doing that, we reduce the chance that false memories are made when the analyte is not seen. So just to remind you, the ribosome binding site determines the protein that's being made. So if you change the sequence, you can reduce the amount of protein that's being made. And this depends on the particular organism. This will come into play in the next couple of slides. So for the next problem, what we do is by removing fluorescence and going blind, just using qPCR, we can reduce any burden that the cells that are carrying the memory have. So hopefully this should alleviate any effect of going back into the off state. And for reducing the chance of any mutations, what we do is we minimize known sequence repeats in the DNA that could cause mutations. And also we remove any non-functional parts in the sensor. Usually what people do is you grab plasmids from lab mates and then you try to stitch them together. By doing that, you carry a lot of unnecessary segments of DNA. So I've done a more bottom-up kind of an assembly where I minimize any non-functional DNA that I have. So by doing both, all three of those changes, I made 3 new designs now. So one of them has fluorescence. So this one I am expecting it to fail because it still has a burden. So here what happens is I flip the promoter region. So in the off state, the fluorescence is not made, and in the on state, the fluorescence gets turned on. And for the second iteration, I remove the fluorescence protein here, and I just put dummy non-coding regions. So in the off and on state, the cell doesn't do anything new, but you can only detect this with qPCR. And for the third device, which is called Frugal, what we do here is we take the full sensor device that's not shown in the first 2 figures and put it between the segments that actually get deleted. So once the deletion happens, this final device does not sense any more AHLs, and it does not make any more integrals as well. So it's the most frugal device that you could make. So what we do now is we put all 3 of these devices into first E. coli and then into other organisms and see how they perform. So as expected, the E. coli data here shows, again, this is a fraction of plasmid that is in the on state. If you get the bacteria to make fluorescence, they don't like it, so they go away over time. But if you have a silent device that's making only a DNA modification, the memory tends to stay alive for a long time. And I've tested this over 8 days, so at least over a week, it seems to be stable. So the conclusion here is that the fluorescence is the main thing that was causing memory loss, and the other modifications are made, helped a little bit maybe, but both the silent and frugal versions are both stable. So the eventual goal we have though is not to test things in lab organisms. We want to test things within wastewater. And if I take a lab engineered E. coli and we put it into wastewater, we don't expect it to survive very well because we're not feeding it with all the amount of nutrients that it needs. So it has to compete with all the existing organisms in the wastewater as well. So the ideal thing would be to grab organisms from wastewater, engineer them, and then put them back and see if they work or not. So that's what I've done here. I brought wastewater into the lab and I've isolated individual organisms from wastewater. And then I transform the memory device, the DNA, into the wastewater organisms through this process called conjugation. And then I do the same assay that I've done for E. coli to see if it works or not. And this is what the data shows. On the top panel, I'm showing E. coli. The bottom is the new organism I've tested. It's one of the Pseudomonas genus from wastewater. I have more organisms, but the data does not look very conclusive, so it's hidden away somewhere else. So the conclusion that we see here is that the wastewater isolate has a slower memory loss compared to E. coli. It seems to stay good for at least like 5 days, I would say. The E. coli is very quick to decay although. So the reason for this is wastewater organisms are known to survive well in low nutrient environments and they grow very slowly. So because of the difference in cell growth, we see this benefit of a longer memory. But we also have two more devices that do not have this memory loss. So if you look at the silent device, E. coli has a very good signal. There's a tiny loss, but it's not significant. But the wastewater device, it seems like whether I expose them to the sensor or not, they have a very comparable signal. And the reason for this is the wastewater might have already flamed for some reason. And when I started the experiment, all of them were already non-state. So this was not really useful. So now if you look at the last sensor that we have, the E. coli has a similar kind of behavior. The on state stays on. In the wastewater though, the on state stays on, there's a slight decay, but when you don't expose them to the sensor, they do seem to gradually be increasing over time. So this could be because either the wastewater organism I picked makes its own AHL, or that there's a higher baseline recombinase activity in the wastewater organism. So because I optimized my sensor for E. coli, I did not optimize it for the other organism. So there could be a discrepancy because of that. So to conclude the first section on DNA memory, I've shown that by using recombinase enzymes, I can record information about biosensing that the bacteria sees into its DNA. And I've shown that this is stable for at least a week to 8 days. And the thing would be for someone to take this and deploy it in environments that it really be useful in, that are inaccessible to normal real-time sensors. Also, further improvements are needed before it actually works in wastewater organisms. So because I've optimized it for E. coli, I would also need to optimize it for the final organism it's going to be in, or think of some other methods where it would work in more than one organism at the same time. So before going to the next section though, what I've done here is, one of the things is I'm trying to use bacteria from wastewater as a sensor. So for that, I had to engineer them by putting in the synthetic DNA that I made. So to do this, this is a very slow process. You have to take one by one organism at a time and engineer them. The alternative would be to grab the whole sample from wastewater and then try to put the device into the wastewater and see as many organisms as it can get into. So the goal here is to engineer the whole community without isolating each organism at a time. So with this goal, what we have is once we put the device into the community, not all of the organisms would take it up. So one challenge we have is how would we figure out which organism within the community has taken up the engineered device and which organisms have not. So for this, we use another sensor which uses RNA memory. So this is my next chapter. And here I'm posing this as a way of storing information across a microbial community because I'm putting it into not isolated organisms, but into a mixed community with many varieties of organisms in. So briefly, horizontal gene transfer is this process that I've used to put my memory sensor into wastewater organisms. So the process is called conjugation, where an organism makes a tunnel and it shares its DNA with its neighbors as a way of helping them out when there's a stress or anything in the environment. So it's good for us because we can engineer communities using this method, but it's bad for public health because if bacteria are freely able to share their DNA, they can share elements that make them resistant to antibiotics, and that would create these super bugs, which are very excited when they see antibiotics because it doesn't affect them at all. So by understanding this process in wastewater, we can use it either for engineering wastewater organisms or to use it as a way of understanding this natural process happening within wastewater as well. So how do people currently do this? People use fluorescence within the bacteria as a way of monitoring which organism they end up in. So if you think about it as information flow, the DNA that we put into the community is carrying information. So each organism that holds the plasmid DNA has the information that the DNA is present in it. But the way that the information is being read out by people is the plasmid DNA makes a fluorescence, and then you sort the cells based on the fluorescence. The ones that are making fluorescence have the device, the ones that don't have not taken up the engineered DNA. So you sort the cells that have the device, and then you sequence their 16S ribosomal RNA to figure out which organisms have taken up the DNA. So the bottleneck in this whole process is that the sorting step takes a long time, especially for samples that are very dilute. It could take anywhere up to 24 hours per sample. And also it can process a very low throughput of samples at a time. So you have to really dilute wastewater before you're able to get through this machine without clogging it up. So what I propose here with this RNA memory is that we can bypass this bottleneck. Instead of storing the memory as the expression of a fluorescent protein, we can store it as a DNA RNA modification. So just by using our final readout method, we can directly figure out which organisms have the DNA in them and which organisms have not taken up the DNA. So just a brief overview of how the 16S ribosomal RNA works. There's this RNA is required for the bacteria to make proteins. So it's there in all the organisms, but there's a slight sequence variation within each organism that enables us to uniquely identify every organism in the community. So that's the way we are using currently, to use these two primers to amplify the RNA within the community and then figure out which organisms there are. So if you think about it in the COVID sense, we can figure out which COVID variant there is, Delta, Alpha, or others, by looking at the sequence that you're able to amplify. So what we do now is, by adding this tiny blue segment onto all the bacteria, we can change the way we are sequencing just by changing the one of the primers to get the exact same output. But we know both which organism there is because of the variable region here. And we also know which organisms are carrying the extra memory that we have added onto them. And the advantages of doing this, as I mentioned before, there's no bottleneck of fluorescence sorting. So it's higher throughput. And also these organisms that need to be identified by this method don't need to make any protein. And reminding you of the central dogma again, DNA becomes an RNA, which is what is causing this output. But then the RNA becomes a protein that would make it fluorescent. So by doing one less step, we are able to identify more of the organisms because their regulatory compatibility might not make them produce enough of the protein. So the tool we use for this is another enzyme. It's called a ribozyme, and this is not a protein, but it's an RNA itself that acts as an enzyme. So the way it works naturally is that there's a whole large segment of RNA, and only the light blue and the light green regions are functional. So the part in the middle splices itself out by doing this cutting and pasting reaction, which was the same as the DNA memory we've seen. So it cuts it in two segments and it joins them together to form this final segment. So what we do is we take this and we modify it such that it's not splicing within a single molecule, but it brings another molecule close to itself, which is the 16S RNA that we are going to target. And then it attaches the barcode onto the 16S RNA to make this hybrid molecule that we are able to sequence very easily. So by using this ribozyme, what I've done is I've put it into not just E. coli, but a few other organisms. And I've tried to see if it works in all the organisms and if the signal is detectable or not. So you can see that the sensor device was engineered for E. coli, but it doesn't work as well in other organisms. But no matter how much more or less of the ribozyme that I express, I'm still able to detect this hybrid molecule that forms by the catalytic action of the ribozyme. So one thing that's interesting is the more of the ribozyme that gets made, the more of these molecules that we can detect. So this is what we're going to use when we put this into a whole community. So if you normalize the signal on the y-axis by the x-axis, we can get like an estimate of how well the sensor works in these organisms. One key thing about the qPCR method is if there's a community that you have tried to engineer, you could have a scenario where it fails completely, where you have no organism that has taken up your device, or there's a few organisms or all the organisms have taken up your device. And by just using this qPCR and by reading out what's on the y-axis, we can tell which scenario of these we are in. So we can predict the frequency of gene transfer without doing more elaborate methods using flow cytometry. So now we take this device and put it not just in isolated organisms, but in a real wastewater community, which has a lot of organisms mixed together. So we take the wastewater community from a local place in Houston. We add the device into that. And then we try to figure out which organisms have taken up the plasmid and they have barcoded their own RNA. So what we are expecting is there is a fraction of organisms within the community that have taken up. Okay, I've come prepared. So there's a fraction of organisms that would have taken up, and we're trying to identify which organisms there are. So by doing two different sets of sequencing, the one on the top, by using the natural way we sequence, we can figure out all the organisms in the community, and by changing the primer on the right to bind only to the barcode, we can figure out the subset of them that have taken up the plasmid and have attached the blue barcode onto their RNA. So we try to apply this in a real wastewater sample now, and we are reading out with these primers for the barcoding. So what we notice here is we initially made a tree of all the organisms that we could find in wastewater. These are all colored in black. Superimposed on that is the organisms that have the barcode on them that are colored in orange here. So you can see that a lot of organisms seem to have taken up the plasmid and have attached the barcode onto themselves. And these organisms are not just closely related to the donor E. coli, but also very farther away as well. The one thing that's not visible in this graph is that there are 20 taxa that are so rare that we could not really detect them within the unmodified 16S. But when the ones that have become barcoded are sensitive enough that they can be detected now. So these organisms are so rare that we could never detect them with flow cytometry or any other method. So this talks to the exquisite sensitivity of this method. And also the interesting thing is there's a lot of organisms that have acquired the barcode. So 50% of all the organisms we see here have taken this up. And also they are distributed among a wide phylogenetic order. So they don't all belong to very closely related organisms to E. coli, which is in the green here. So this graph on the x-axis shows the fraction of organisms that are within the order that have been barcoded. So a few of them have more than 60% that have been barcoded, but it's also, there's a major bulk distributed across 6 different taxa. So they span 6 different taxonomic orders. So to summarize the RNA part, we showed that a ribozyme can be used to modify the 16S RNA as a way of recording memories. And then we can use this as a device to track which organisms within the community have taken up your plasmid DNA as a way of engineering community. And I've shown that this is more, I haven't shown it, but I believe that it will be more sensitive than the current fluorescence-based sorting, especially because we saw 20 taxa that are very rare in the community. And this is also broad host enough that it works in a bunch of organisms without requiring fine-tuning of the sensor. And in the future, we can deploy this to identify organisms that respond to particular environmental changes, depending on when the promoter that's guiding the tool is getting activated. So we can mix the paradigm of the DNA memory with this to make a hybrid device. So to summarize, the DNA memory is good for long duration, and I've shown that it's stable for 1 week, and it works very well in inaccessible and opaque environments. So the future directions that this needs is that when we put it into other organisms that are not lab optimized, we need to find a way to optimize them for each individual organism or to find a universal optimization strategy. And for the RNA section, we can use the 16S RNA as the medium to store the memory. And we can use it to monitor the genes being transferred around in a community. And it works in a broad host range of taxa and within a very complex microbiome like the wastewater. And we can use this to identify organisms that respond to other environmental conditions as well. And I would like to acknowledge both my advisors. It has been fun working with two different mindsets at the same time. It's like a yin and a yang. Geoff has a lot of very broad set of ideas, and Lauren likes to narrow them down to where they fit the project. And I also want to thank my committee for all the valuable feedback that I've got from them. And I've been part of two different labs, the Lawrence Stadler lab and the Silberg lab. And it's been a great joy working with so many people in both labs and mentoring them and carrying knowledge, best practices across labs, putting them into YouTube videos or all possible ways. And also the RNA memory work was a part of a collaborative effort. And the concept was driven by people from James Chappell Lab. And I would also like to thank all the friends that I've had at RISE who made my time here very memorable. And also my parents, without whom I would not have started my PhD, and my partner Shweta, whose photo, she doesn't want to be here. She helped a lot coping with all the stress of grad school. And with that, I'll leave you with the summary slide in case you have any questions.
Comparing the silent and the Google one in July. Do you have an idea why like the silent one ended up being better, or it looks like it was better than the Google one?
Yeah, that is a good question. Yeah, so the point here is that it was just unfortunate that the 3 colonies that I decided to pick for my assay were all already flipped. But if that didn't happen to be the case, then I think both of them would have been comparable. So the point is that this sensor is irreversible. So once the memory turns into a non-state, if you happen to pick the wrong colonies, then they are not useful anymore. But other than that, I believe that both these devices should work similarly. Any question?
How modular is long-term small sensor?
Yeah, so I've sensed both the AHL molecule that I was talking about and also another sugar called arabinose. There was a slide in the middle. If anyone noticed the x-axis, it was not AHL. So you can sense multiple things with it, and the sensor is quite plug and play. So all you need is to be able to tune it so that the leak of the recombinase doesn't overwhelm your system.
Big offers. Question about the time scale of their RNA-based sensors. Have you tried doing like 9 courses across wastewater community to see if sort of signals are coming in, and it's RNA editing, so probably from here. And do you see any signal coming through? So notice you have a bunch of taxa with like one community thing. I'm wondering if like there were more along those taxa things just like that went to the time.
Yeah, we plan to do that, but we did not do it for the first paper. Along with the same lines, we also characterized how stable the RNA is. So we made the RNA memory turn on, and then we arrested the translation, and then we transcription, sorry. And we try to figure out how long the RNA lasts for, and we find that the half-life is around like 2 minutes or so. So it's pretty short-lived once we take it out of the cell. So it will be useful for the dynamic measurement, like you said. If you follow the same process along many days within wastewater, the information is going to be very useful. So there's a question from the chat. I'm wondering what you think are the major reasons that some of the wastewater microbes didn't get barcoded. So this could be two things. They either did not take up the plasmid that we try to. Yeah, so when we do the gene transfer into a community, which organism gets it depends on the donor as well. So if it's an E. coli, it will try to give it to the ones closest to E. coli. It also depends on the plasmid backbone. So the method that we have developed is so easy that now someone else can take these, change the donor, change the plasmid, and then test out which subset of organisms changes based on that. So I feel like it's not a bottleneck of the RNA memory itself, but it's which organisms that can take up the DNA is more upstream, the bottleneck. Okay, so the question on chat is it possible to have the transferred gene get lost during bacterial multiplication? Yeah, so usually the plasmid that we give to the community is compatible with a broad range of organisms. So the organism tries to keep it alive within itself. But if you change the device, if you change the origin of replication, you can see a different set of organisms getting the plasmid. But we can also test a kind of a plasmid that does not have an origin to see that, because that's not geared for keeping the device within the organism. We could see whether how quickly the plasmid is lost within the community. That would be an interesting idea to do as well. And are there any regulatory barriers to real life application? Yeah, I think we can't directly put engineered organisms into the environment, even though that's what I've alluded to in the introduction. So this is a very long-term thing, and there's a lot of safety aspects that need to be taken into account. So the RNA device is being used by another student in the lab, and what he's trying to do is to use it as a way to screen a promoter library within a diversity of organisms. So usually when we do promoter libraries, we pick a single organism. But with this method, you get both the taxonomy information as well as how well the device is working based on how many reads you get. So you can use it as that. You could also hook it up to a conditional promoter and see whether you can make you can record a subset of community that has the plasmid and another subset of that community that turns on the promoter. Then you give it a certain condition.
So thank you, Prashant, one more time.